So this paper is also by Giles Van de Waal. He has a different team this time. I don't recognize the names from the first paper, so it's from 2007. So it's a little older, but it's right along the lines of what you were interested in. Brain responses to violet, blue, and green monochromatic light exposures in humans. Prominent role of blue light and the brainstem. That's the title. So it seemed like right up your alley. There's some background. Relatively long duration retinal light exposure elicits non-visual responses in humans, including modulation of alertness and cognition. These responses are thought to be mediated in part by melanopsin-expressing retinal ganglion cells, which are more sensitive to blue light than violet or green light. Just, that's the melatonin that we were talking about before. Green light. The contribution of the melanopsin system and the brain mechanisms involved in the establishment of such responses to light remain to be established. Methodology, principal findings. We exposed 15 participants to short duration, 50-second monochromatic violet, 430 nanometers, blue, 473 nanometers, and green light onset. And green 527 nanometer light exposures of equal photon flux, 10 to the 13th or centimeters squared per second, while they were performing a working memory task in fMRI. At light onset, blue light as compared to green light increased activity in the left hippocampus, left thalamus, and right amygdala. During the task, blue light as compared to violet light increased activity in the left middle frontal gyrus, left thalamus, and bilateral area of the brainstem, consistent with activation of the locus coeruleus. Conclusion: significance. These results support a prominent contribution of melanopsin expressing retinal ganglion cells to brain responses to light within the very first seconds of an exposure. The results also demonstrate the implication of the brainstem in mediating these responses in humans, and speak for a broad involvement of light in the regulation of brain function. Light processing has been studied extensively in the context of circadian biology, with emphasis non-visual or non-image forming effects of environmental light irradiance. These non-visual effects include the desynchronization of the circadian system, suppression of melatonin, regulation of sleep, as well as improvements of alertness and cognition. 106. We have shown that non-visual responses related to alertness and cognition are associated with changes in regional brain activity detected by positron emission tomography, PET, and functional magnetic resonance imaging, fMRI, 7 through 9. A number of recent studies using a wide variety of methodologies revealed that acute or longer-term human non-visual responses are most sensitive to monochromatic light of wavelength between 460 and 480 nanometers. 2 through 6, 9 through 14. This sensitivity is much shorter than the overall maximum sensitivity of the phototopic system, approximately 550 nanometers. Overall maximum sensitivity. And does not coincide with the maximum sensitivity of any of the individual classical photoreceptors. Rods 505 nanometers, S cones 430 nanometers, M cones approximately 530 nanometers, L cones 560 nanometers, 15 through 16. So it's saying that it's non-visual light that is, the sensitivity is much shorter than the overall maximum. So it's not necessarily all of it being visual light. Some of it is, but not all of it. A fifth retinal photopigment, melanopsin, was recently discovered and shown to be expressed in retinal ganglion cells, RGC, that are intrinsically light sensitive. 18. With a maximum sensitivity between 420 and 480 nanometers, 1921. And melanospin expressing RGC are implicated in non-visual responses to light, 18 through 22. They project numerous brain structures in rodents, 23 and 24, including hypothalamic nuclei such as the suprachiasmatic, so there's that word again, suprachiasmatic nucleus, SCN, and the ventrolateral preoptic area, VLPO. We've read the suprachiasmatic definition a couple times, but let's do it one more time just because we keep running into this word. And so by now, I'd really never run into it until we started reading these papers. A pair of neuron clusters in the hypothalamus situated directly above the optic chiasma that receives photic input from the retina via the optic nerve and that regulate the body's circadian rhythms. Let's look up chiasma. The chiasma, the X-shaped structure formed at the point below the brain where the two optic nerves cross over each other. So it's where the optic nerves cross over each other, and the suprachiasmatic cluster or nucleus is a pair of neuron clusters in the hypothalamus that basically oversee and regulate the body's circadian rhythm. So yeah, that's why we keep reading on papers about circadian rhythm. So that's why we keep seeing these nerve clusters mentioned, as well as many non-hypothalamic structures, including the olivary pretectal nucleus, OPN, and the amygdala. Melanospin expressing RGC also project to areas typically involved in vision, such as the lateral geniculate nucleus and the superior colliculus. In addition, melanospin expressing RGC project to the LGN and OPN in Machus. 25. These neuroanatomical pathways provide a mechanism by which irradiance changes could affect many brain functions, i.e. circadian entrainment, pupillary constriction, arousal, attention, and emotion regulation, as well as vision. 2, 4, 8, 10, 13, 25, and 26. However, classical visual photoreceptors are necessary to induce complete non-visual responses to light. 27. In addition, RGC, which do not express melanospin and presumably are not intrinsically photosensitive, project to the SCN intergeniculate nuclei, IGL, of the thalamus and the VLPO, suggesting that signal arising from the classical retinal photoreceptors reaches these structures. 24 and 28. The relative contribution of the different retinal photoreceptors has not been fully assessed. Rod and cone responses to light are typically time-locked to the exposure, i.e. responses start and cease within a few milliseconds after light is turned on and off, respectively. In addition, quick attenuation of rod and cone signals occurs in the presence of a constant light stimulus. 25. Intrinsic light responses of the melanomus personic expressing RGC are much more sluggish and do not show attenuation. They are only detected seconds after light onset, and firing is maintained for minutes after the end of the light exposure. This feature suggests that these cells are able to account for the long integration time of the non-visual system, 18 to 25. However, melanospin expressing RGC receive intrinsic inputs from rods and the three classes of cones, which enable melanospin expressing RGC to instantaneously respond to light exposure and suggest an important role for rods and cones in the non-visual response to light early in the exposure. 25. Accordingly, assessment of the relative efficacy of different wavelengths indicates that the M cones contribute importantly to the initiation of the response in rodents, but later the melanospin expressing RGC are the dominant contributor 13. Similarly, at lower irradiance, classical photoreceptors are sufficient to drive pupillary constriction in rodents, while at higher irradiance, melanospin expressing RGC are required to induce a full response 29. In addition, the wavelength sensitivity of rat SCN neural responses to light flashes suggests a contribution of rods and all cones to brief light exposures 30. A role for S cones in non-visual responses in humans, as was inferred from data showing a greater increase in subjective alertness under violet light exposure, 420 to 440 nanometers. 31. However, most human studies investigating the mechanisms for non-visual responses to light employed monochromatic exposure stimulating most melanospin expressing RGC or M and L cones, but not S cones 2, 4, 5, and 9. S cone contribution to non-visual responses to light using ultraviolet light preferentially triggering these photoreceptors remains to be firmly established. In addition, non-visual responses to different wavelengths in humans have only been characterized after relatively long duration exposures, at least tens of minutes. Presumably after a substantial attenuation of rod cone signals, thus the relative contributions of blue and violet, green lights, and by inference of melanospin expressing RGC, S, and M cones, and the establishment of non-visual responses to light have not been assessed in humans. Besides the known projections of melanospin expressing and non-melanospin expressing RGC to brain structures involved in non-visual functions, most of the brain mechanisms and pathways mediating non-visual responses to light exposure are unknown. In rodents, the SCN and thalamic IGL receive light irradiance information almost directly and appear, therefore, to be strongly implicated in eliciting non-visual responses to light. 32 and 33. The SCN and IGL project to many brain structures involved in arousal regulation, and a functional indirect connection between the SCN to the brainstem locus coeruleus LC has been established. 35. This SCN brainstem projection may be the pathway by which light modulates alertness. However, beyond these candidate subcortical and brainstem structures, the brain mechanisms involved in generating physiological and behavior non-visual responses to light have not been characterized in animals. In humans, using PET and fMRI, we have identified neural correlates of the alerting effects of a bright white light exposure greater than 7,000 lux delivered at night or during the day in brain areas such as the intrapartial sucleus IPS, hippocampus, thalamic pulvinar, insula, and hypothalamus 7 through 8. More recently, we demonstrated that brain activity related to a working memory task is maintained or even increased by blue 470 nanometers monochromatic light exposure, whereas it decreases under green 550 nanometer monochromatic light exposure 9. These effects were detected in areas implicated in working memory such as the thalamus, the insula, IPS, and supramarginal gyrus. The middle frontal gyrus, MFG. However, these studies were carried out using relatively prolonged light exposures, 17 to 21 minutes. The brain areas first affected by light exposure and by inference involved in establishing non-visual responses to light are therefore largely unknown in humans. In the present study, we use fMRI to specifically assess early effects of light over the entire brain while participants were performing an auditory working memory task. We used alternating violet 430 nanometer, blue 473 nanometer, or green 527 nanometer monochromatic light exposures of equal photon density to investigate the processing of stimuli preferentially triggering S cones, melanospin expressing RGC or M cones, respectively. Light exposures lasted 50 seconds, a very short duration from a human circadian biology perspective. We hypothesized that such short duration exposures would induce sustained modulation of the brain responses related to the blocks of the task performed, and that these modulations were wavelength dependent. This would allow insight in the relative contributions of the different retinal photoreceptors early on in the establishment of non-visual responses to light. On such a short time scale, it is difficult to establish whether the detected brain activity modulations constitute non-visual or visual responses. This, however, is not essential for our aim, which was to identify brain mechanisms involved in establishing responses to light exposures, which eventually will lead to non-visual responses such as changes in cognition and alertness. We also hypothesized that such short exposures would not induce wavelength-specific responses in a large number of brain areas, but would mainly affect a few areas involved in the establishment of the responses, presumably subcortical and brainstem areas. The results support our hypothesis and suggest a prominent role of melanospin expressing RGC in the establishment of brain responses to light. Methods. Subjects. Participants were healthy, young subjects, N equals 15, 8 females age 19 through 27, median 22, BMI 18.7, 27.3, median 22.2. A semi-structured interview established the absence of medical, traumatic, psychiatric, or sleep disorders. Absence of color blindness was assessed by the 38-plate edition of Aishira's test for color blindness, Kanehara Shupman Company, Tokyo, Japan. All participants were non-smokers, moderate caffeine and alcohol consumers, and were not on medication. None had worked on night shifts during the last year or traveled through more than one time zone during the last 2 months. Extreme morning and evening types, as assessed by the Horn-Ostberg Questionnaire 36, were not included. None complained of excessive daytime sleepiness as assessed by the Epworth Sleepiness Scale 37, or of sleep disturbances as determined by the Pittsburgh Sleep Quality Index Questionnaire 38. All participants had normal scores on the 21-item Beck Anxiety Inventory 39 and the 21-item Beck Depression Inventory 40. They were right-handed as indicated by the Edinburgh Inventory 41. Participants gave their written informed consent and received a financial compensation for their participation. The study was approved by the Ethics Committee of the Faculty of Medicine of the University of Liège. So that's, again, a study that has been approved by a ethics committee. This time by the University of Liège, but any sleep study or anything like that that can involve human testing has to or should go through an ethics committee. Volunteers followed a regular sleep schedule during the 7-day period preceding the laboratory segment of the experiment. Compliance to the schedule was assessed using wrist actigraphy, ActiveWatch, Cambridge Neuroscience UK, and sleep diaries. In order to record 2 volunteers on the same day at approximately the same circadian time, volunteers were requested to follow one of 2 sleep schedules differing by 1.5 hours, 2300 to 07 hours, plus or minus 30 minutes, or 30 hours to 0830 hours, plus or minus 30 minutes. Volunteers were requested to refrain from all caffeine and alcohol-containing beverages and intense physical activity for 3 days before participating in the study. Subjects were first maintained in dim light, less than 5 lux, for 2 hours and then scanned during 3 consecutive 20-minute sessions. 3 drops of tropicadium, 0.5% tropicol, were administered in the eyes 20 minutes before entering the scanner to inhibit pupillary constriction. In each session, subjects were alternatively exposed to monochromatic 50-second light exposure separated by 5 to 14 periods of darkness, less than 0.01 lux. Figure 1b. Monochromatic light was violet, 430 nanometers, blue, 473 nanometers, or green, 527 nanometers, and aimed primarily at S cones. Mellon spin expressing RGC and M cones respectively. In each session, 2 wavelengths were presented and alternated. Each color was presented 10 times per session. Subjects were exposed to the 3 possible combinations of wavelengths over the 3 sessions. The order of the combinations and the wavelengths of the first light exposure in each combination were counterbalanced over subjects. Supplemental table S1. In accordance with other protocols in this research area, the photon density of all light exposures were identical to allow the assessment of the relative contribution of the photoreceptors most sensitive to each wavelength. Photon density was set at 10 to the 13th photon centimeters squared per second. Because at this level, non-visual responses at night and during the day depend on the wavelength of the light exposure, 2 through 6, 9, 11, 12, and 42. This photon density was equivalent to the illumination level of 4, 7.5, and 24.5 photopic lux for violet, blue, and green light exposure, respectively. The first light exposure occurred approximately 4 hours after habitual wake-up time, i.e. during the biological day when melatonin secretion is low. 1. During each session, participants performed an auditory 2-back working memory task, 43, which is reliable, executed by a majority of subjects and does not explicitly depend on visual input. Subjective alertness scores, as assessed by the Karlonsky Sleepiness Scale, KSS 44, were collected every 30 minutes during the 2-hour preparatory period and between each session while in the scanner. During the data acquisition period, all subjects interacted with the same investigator who used a standardized set of sentences between each session. This protocol was implemented in order to minimize variation in motivational state due to social interactions, e.g. encouragement by an investigator, which may modify brain responses. 45. No feedback was given on performance. Volunteers were trained on a shortened version of the protocol and habituated to the experimental conditions at least a week before the experiment. Subjects had to reach 75% of correct responses on the 2-back test at the end of the training to participate to the experiment. 2-back task. Stimuli consisted of 9 French monosyllabic consonants that were phonologically different so that they could easily be identified. Stimuli were 500 milliseconds long and interstimulus interval was 250 milliseconds. For each consonant, volunteers were requested to state whether or not it was identical to the consonant presented to stimuli earlier by pressing a button on an MR-compatible keypad for yes and another one for no. Series of stimuli were constructed with 30% positive answers. 14 consonants were presented in each elimination period and for a total of 35 seconds, and 2 to 5 consonants were presented in half of the darkness periods for a total of 5 to 12.5 seconds. Series could therefore be 33 consonants long if a darkness period with a task was placed between 2 consecutive elimination periods where the task was performed. Series were presented only once and were randomly assigned to one of the scanning sessions. Rest periods could last up to 44 seconds if a rest period in darkness was placed between 2 consecutive illumination rest periods. Stimuli were produced using Cogent 2000. It's got a website. Implemented in MATLAB, MathWorks Incorporated. I've used MATLAB. That was a long time ago, though. On a 2.8 gigahertz Xenon Dell personal computer, Round Rock, Texas, and were transmitted to the subjects using MR control amplifier and headphones, MR Confon, Germany. The first session was preceded by a short session during which volunteers had to set the volume level to ensure an optimal auditory perception during scanning. Narrow interference bandpass filters, full width at half maximum FWHM 10 nanometer Edmond Optic UK were used to produce the three monochromatic illuminations. A filter wheel, AB301T Spectral Products NM, was computer controlled to switch bandpass filters and thereby change light wavelength. The light was transmitted by a metal-free optic fiber from a source PL900 Dolon Jenner Industries MA to two small diffusers placed in front of the subject's eyes. The diffusers were designed for the purpose of this study and ensured a uniform illumination over the entire visual field. Light was administered through a 4 by 5.5 centimeter frame placed 3 centimeters away from the eye. The spectra of each monochromatic light were checked at the level of the diffusers. Avaspec 2048 Avantis at the Netherlands, and the 430 nanometer, 480 nanometer, and 532 nanometer bandpass filters produced light with a maximum radiance at respectively 430.3 nanometers, 472.8 nanometers, and 527.3 nanometers. Irradiance could not be measured directly in the magnet, but the light source was calibrated and irradiance estimated to be 10 to 13 photons per centimeter squared per second. 840C power meter, Newport, Irving, California. After pre-receptoral lens absorption for the different wavelengths was taken into account, 46, the total amount of light received during the experiment was well below the blue light hazard threshold. In order to uncorrelate task and light onsets, the auditory task was performed during 35 seconds of the 50-second illumination periods. Half of the illumination started with 15 seconds of rest, the other half terminated with 15 seconds rest periods. In addition, a 0 to 1 second jitter was implemented between light onset offset and task onset offset when they occurred simultaneously in order to further uncorrelate them. Darkness periods, less than 0.01 lux, separated all 50-second illuminations. The auditory task was performed during half of the darkness periods, and the duration of which were then 5 to 12.5 seconds. Rest was requested during the other half, in which case darkness was lasting 9 to 14 seconds. The illuminations with one color were always followed by darkness periods, then by illuminations in the other color of the session. Behavioral data analysis. Accuracy scores were always very high, so we computed D prime and criterion values following the signal detection theory in order to identify possible changes in behavior not reflected in overall accuracy. Repeated measure ANOVA, ANOVA, with light condition and session as within subject factors were carried out separately on D prime, criterion, and reaction times. Repeated measures ANOVA with repetition as within subject factor were computed on subjective sleepiness scores. All behavioral analysis were computed with statistics. Statistica 6.1. StatSoft France. Functional MRI. Data acquisition. Functional MRI time series were acquired using a 3T MR scanner. Allegra, Siemens, Germany. Multi-slice T2 weighted fMRI images were obtained with a gradient echo planar sequence using axial slice orientation. So it's just telling you the angle at which the scanner is scanning. That's 32 slices, voxel size 3.4 by 3.4 by 3 millimeters cubed, because it's a volume. That's why it's cubed. Matrix size 64 by 64 by 632. Repetition time equals 2130 milliseconds. Echo time 40 milliseconds. Flip angle 90 degrees. The 4 initial scans were discarded to allow for magnetic saturation effects. There was little variation in the number of scans per session. Blue-green sessions 563.3 plus or minus 5.9 mean plus or minus SD. violet-blue sessions, 563.4 plus or minus 6.2. Green-violet sessions, 563.3 plus or minus 7.5. Head movements were minimized using a vacuum cushion. A structural T1 weighted 3D MP Rage sequence TR 1960 millisecond, TE 4.43 millisecond, TI 1100 millisecond, FOV 230 by 133 centimeters squared, matrix size 256 by 256 by 176, voxel size 0.9 by 0.9 by 0.9 millimeters, was also acquired in all subjects. Wow. So there's scanning the subjects with the fMRI, and it's just telling you the angles and how they're doing it and how they're weighting the sequence. That's all that was. Functional, just telling you how they got the MRI data acquisition. Now they're going to talk about how they analyzed the data that they gathered. Functional MRI data analysis. Functional volumes were analyzed using statistical parametric mapping 5. So a program. It's got a website. Implemented in MATLAB, another it's a math program for mapping. You can map 2-dimensional and 3-dimensional curves. It's not as used as much anymore, but yeah, back in the early 2000s, when I first took calculus, it was big stuff. OCCC was very thrilled that they had a copy and that they could show people how to use it. In my calculus class, we had a lab, and it was MATLAB, and that was all you did was map things. It was a very powerful tool, and it was just as when these programs started to really take off, producing visual confirmation of formulas and equations and things like that. So it was very useful, and obviously it was used even in like these sleep studies. So it had a very, very wide applicability. They were corrected for head motion, spatially normalized standard SPM5 parameters to an echoplanar imaging template conforming to the Montreal Neurological Institute MNI space, and spatially smoothed with a Gaussian kernel of 8mm FWHM. The analysis of fMRI data based on a mixed effects model was conducted in 2 serial steps, accounting respectively for fixed and random effects. For each subject, changes in brain regional responses were estimated using a general linear model in which the different parts of the experimental design were modeled using either boxcar or stick functions convolved with a canonical hyodymic response function. Boxcar functions modeled the 15-second brain or 15-second rest illumination periods and the 35-second illumination periods, including the 2-back task and the darkness periods during which the task was performed. Stick functions modeled light onsets and light offsets. Nominal spin expressing RGC do not cease firing at light offset 25. So brain responses to light offsets are unlikely to represent a non-visual response to light. Further, rest periods during the illuminations were short as compared to the task periods and were contaminated by the performance of the task. The regressors modeling offsets and rest periods were therefore considered as covariates of no interest, together with movement parameters derived from realignment of the functional volumes. High-pass filtering was implemented in the matrix design using a cutoff period of 256 seconds to remove low-frequency drifts from the time series. Serial correlations in the fMRI signal were estimated using an autoregressive order 1 plus white noise model and a restricted maximum likelihood algorithm. The effects of interest were then tested by linear contrast generating statistical parametric maps. The summary statistic images resulting from these different contrasts were then further smoothed 6mm FWHM Gaussian kernel and entered in a second level analysis. This second step accounts for intersubject variance in the main effects of light condition, random effects model, and corresponds to a one-sample t-test for brain responses to the t-back series and light onsets. The resulting set of voxel values for each contrast instituted maps of the t-statistics threshold at p uncorrected equals 0.001. Statistical inferences were performed after correction for multiple comparisons on small spherical volumes SVC 10mm radius at a threshold of p SVC equals 0.05 around a priori locations of activation. Activations were expected in structures involved in back tasks, arousal regulation, and showing non-visual responses to light in our own fMRI and PET work. Brain areas to which the melanospin expressing RGC project were functionality linked to the SCN were also considered as a priori locations of activation. Standard stereotactic coordinates of previously published a priori locations used for SVC are as follows: amygdala 22, -6, -15, 49. Hippocampus -30, -30, -2, 8. LGN -23, -21, -3, 15. LC 2, -32, -20, 51. Thalamus -14, -14, -16, 9. Results. Behavior. All sessions and light conditions were identical from a behavioral point of view. Statistical analysis showed that performance, reaction time, and accuracy was always high and was not affected by the light condition or sessions. Supplemental data S1, figure S1AC. Computation of subjective sleepiness scores revealed that entering the scanner and the associated change in posture significantly increased sleepiness. However, bias associated with variations in sleepiness was prevented by the pseudo-randomization of session types. Supplemental data S1, figure S1DF. fMRI data, sustained effects. The analysis of fMRI data first focused on the brain responses recorded during the blocks of the two-back task. The effects described below are therefore unsustained or are sustained because they describe differences between light conditions that were maintained. For the duration of the blocks, significant differences between violet and blue light exposures were detected in the left MFG and in the left thalamus, a few millimeters away from the location for which we previously found a wavelength-dependent effect of light 9, as well as in two areas of the brainstem. Spatial resolution of the fMRI technique does not allow a precise identification of the brainstem nuclei included in the activation areas, but the location of the activations is compatible with several pontine nuclei involved in arousal regulation, and in particular with the LC bilaterally. Figure 2, Table 1, 52. Activity estimates show figure 2 right panels that compared to the violet light conditions, responses were greater under the blue exposure in these four brain areas. No significant differences between blue and green light exposures and between violet and green light exposures were detected during test periods. Supplemental tables S2. Additional analysis suggested that the differences between light conditions were stable during the 50-second illumination. Supplemental method S1, supplemental tables S3. Transient effects. Two monochromatic light exposures were initiated 10 times in each session. This number of events was sufficient to conduct an analysis on the transient brain responses triggered by the onsets of the different light exposures. Significant differences between responses to light and green light onsets were observed in two limbic areas. the left hippocampus and the right amygdala, and in a location in the left thalamus which was identical to that identified in the analysis of the sustained brain responses. Figure 3, Table 2. Activity estimates, Figure 3, right panels, show that these three brain areas strongly responded to blue light onsets, while their activity was barely affected by green light onsets. No significant differences were found between violet and blue light onsets, while violet light onsets were found to increase left LGN activity significantly more than green light onsets. Table 2, Supplemental Figure S2, Supplemental Tables S4. Discussion. This study compared the brain responses elicited by violet, blue, and green monochromatic light exposures of short duration, 50-second and equal irradiance, 10 to the 13th pH, that's photons per centimeter squared per second, and presented in pairs of colors in 3 separate sessions. We hypothesized that these short light exposures would induce wavelength-dependent modulations of brain responses, mainly in subcortical and brainstem areas. Accordingly, we report sustained wavelength sensitivity modulations of the regional brain responses evoked by a working memory task. In particular, blue light is superior to violet light in eliciting these response modulation. These results cannot be accounted for by any measurable difference in alertness or performance, nor by any order of expectation effect. Supplemental data S1. These modulations are considered sustained because the brain activity is continuously enhanced during the 50-second blue light blocks and consistently so during the whole blue-violet fMRI session. Although sustained, these light-induced responses may be considered early responses when compared to the brain responses we reported after 18 minutes of blue monochromatic light exposures 9. As predicted, these early responses primarily involve subcortical areas related to arousal, brainstem, and thalami. At the cortical level, the responses are enhanced in a single area, the MFG. This result contrasts with the enhanced responses in the widespread cortical regions elicited by longer exposures 9, and suggests that the functional recruitment of the cortex requires longer exposures and possibly the activating influence of subcortical substructures. An unexpected finding concerned the transient responses triggered at the onset of light exposures in 2 limbic areas, i.e. the amygdala and hippocampus and the left thalamus. Irrespective of whether the subjects were engaged in the working memory task, these results are remarkable because blue light was superior to green light in eliciting these brain responses, even though illuminance was about 5 times higher for the green light. Collectively, these sustained and transient responses show the efficacy of short wavelength 473 nanometer light in modulating brain activity, and indirectly suggests the involvement of melanospin expressing RGC, which are the photoreceptors most sensitive to this wavelength. Non-visual versus visual responses. This study aimed at identifying brain and retinal mechanisms involved in early responses to light exposure that would likely be implicated in establishing the non-visual responses that have been reported using longer duration exposures 2 through 11, 13, and 14. The experiment was not designed to ascertain whether or not these mechanisms constituted non-visual responses to light. In fact, we believe that the distinction between non-visual and visual responses cannot be made in this experiment. Furthermore, there is growing evidence for a considerable overlap between visual and non-visual photoreception systems. In rodents, rods and cones are involved in the non-visual responses to light, 1327-29, and non-expressing melatonin-expressing RGC project to non-visual brain structures, 24 and 28. On the other hand, melanospin expressing RGC project to project to structures typically involved in vision in rodents and primates, 23-25. Appear to regulate visual processing both in rodents and in humans, 53-54. Therefore, identifying a photoreceptor implicated in a response does not directly inform on the non-visual visual nature of that response. In addition, in our protocol participants' visual system was stimulated during light exposures as participants obviously received the light. We minimize differences in photoreceptor stimulation by equating irradiance level across wavelength. However, because of stimulation of retinal photoreceptors changed with the wavelength of the exposure, signals transmitted to the visual system varied between wavelengths. Finally, we report effects of light at onset and after a few tens of seconds of exposure. From a human circadian biology perspective, this constitutes a very short time scale. During this period, a mixed attenuation of cone signal and increase in the intrinsic response of melanopsin expressing RGC has been observed. 25. The sustained modulations of brain responses related to the task blocks arguably represent non-visual responses. Indeed, we report light-induced modulations of brain responses that are related to an auditory task and are most sensitive to blue light, which suggests the involvement of the melanopsin expressing RGC. In addition, whereas visual responses show quick attenuation, 25, we detected modulations of brain responses that were maintained for 50 seconds. One could also argue that transient responses are non-visual since they are likely to be predominantly mediated by melanopsin expressing RGC. However, non-visual responses are characterized by sustained activity modulation, and the transient effects we detected could therefore be considered to be mediated by the visual system. We believe that a more accurate description of our data is that we detect the transient and sustained brain responses to light that appear to be predominantly mediated in melanopsin expressing RGC without qualification with respect to the visual or non-visual nature of these responses. Just talking about the difference between the non-visual and visual light and how it is perceived and how it reacts. Sustained responses during task performance. A sustained enhancement of responses to the working memory task was observed during the exposures of blue rather than violet light in the brainstem, the thalamus, and the left MFG. No difference in response was observed when contrasting blue with green light or violet with green light. These results suggest that the sustained response modulation of monochromatic light is most sensitive to blue light and least sensitive to violet light. The status of green light cannot be precisely estimated, but it is consistent with the intermediate sensitivity. By inference, these results suggest that melanopsin expressing RGC contribute most of these sustained responses to light. The observed, albeit smaller, responses to violet and green light could be explained in two ways. According to the first interpretation, they represent a combination of a weak contribution of S cones and an intermediate involvement of M cones. In line with this interpretation, melanopsin expressing RGC and M cones 13 seem to contribute greatly to non-visual responses to light during the first minutes of the exposure in rodents. According to the second interpretation, melanopsin expressing RGC are the only photoreceptors involved in the light-induced modulations of brain activity, and the observed smaller responses to violet and green light simply reflect the reduced sensitivity of melanopsin to these wavelengths. Melanopsin would then appear to have a greater sensitivity to wavelengths longer than 473 nanometers, as compared to wavelengths shorter than 473 nanometers. The brainstem area, which was recruited by blue light, corresponds tentatively to the LC. This result is important because it is the first time a brainstem structure is shown to respond to light in humans. The LC may be a key structure in establishing effects of the light. It could receive light information from the SCN, with which it is functionally connected into rodents. 35. As a major source of brain refine, it is in a position to modify the level of arousal. 55, 56. Finally, it is well established that the LC is involved in cognition and executive functioning, in particular. 55. Ophthalmic nuclei appear as the structures most consistently recruited in humans by non-visual responses to light. Polychromatic light, white light exposure 8, monochromatic 470 nanometer, blue light exposure 9. Like the brainstem, the thalamus has a key structure involved in the interaction between alertness and cognition in humans. 57. And it is recruited by working memory tasks. 58. In addition, the thalamus might receive irradiance information through a two-step pathway linking melospin expressing RGC to the superior colliculus, which in turn projects to the pulvinar. 59. Cortical responses were enhanced after recurring 50-second periods of blue relative to violet monochromatic light exposure, only in the left MFG, an area implicated in working memory. 58. This limited recruitment of cortical areas contrasts with our previous experiments, which used longer light exposures. Exposures to white light for 21 minutes enhanced cortical responses to an auditory attention task in widespread cortical areas, dorsolateral prefrontal cortex (IPS), superior parental lobe, insula, precuneus, anterior and posterior cingulate cortices, and superior temporal gyrus. 8. Likewise, 18-minute exposure to monochromatic blue 470 nanometer light as compared to green 550 nanometer light increased the responses induced by working memory task in the left IPS, supramarginal gyrus, MFG, and the right insula. 9. Collectively, these findings suggest that non-visual responses require some time to build up in the cortex. The assessment of this time course will require further studies characterizing the relations between photon density, duration of light exposure, and regional brain responses. Such studies will benefit from the methodological advances presented in this paper, namely within recession assessment of the light-induced brain responses, which provide a fast, reliable technique to characterize light-induced brain responses. Because there are no direct connections between non-visual system and the cortex, we surmise that the light-induced enhancement of cortical responses follows indirect pathways involving activating subcortical structures. An unanticipated result, transient responses to light onsets. An unanticipated result was the responses in left hippocampus, left thalamus, and right amygdala at light onset of blue relative to green light. Such differential response was not observed in the comparison between blue and violet lights, or between violet and green lights. These results are surprising for several reasons, because the visual system is most sensitive to green, 555 nanometer light, 15, and since light onset is a typical visual stimulus, we expected green light to induce the greatest responses at onsets. In addition, M and L cones signals were reported to elicit on responses in melospin expressing RGC, whereas S cones were reported to mediate all OFF responses. 25. Green light should therefore increase activity in these melospin expressing RGC at light onset, and brain responses mediated by melospin expressing RGC at light onset should be at least sensitive to violet light. Taken together, these elements suggest that melospin expressing RGC contribute most of these transient limbic and thalamic responses to light onset. The reduced response sensitivity to violet and green lights could be explained in two ways. The contribution of N cones could be considered to be as the weakest, and involvement of S cones as intermediate, or melospin expressing RGC should be deemed as the only photoreceptors involved, with a greater sensitivity to wavelengths shorter than 473 nanometers as compared to longer than 473 nanometer wavelengths. Both assumptions could therefore suggest a shift in sensitivity between the transient brain responses related to light onsets and the sustained responses associated with to task blocks. Accordingly, shift in wavelength sensitivity with exposure, duration, intensity has been reported for circadian phase shift and pupillary constriction in rodents, 13 and 29. However, in the brain areas showing responses most sensitive to blue light. Changes in wavelength sensitivity are inferred based on comparisons with blue light, not on significant differences between violet and green light. Characterizing duration gradients relationship will provide important data on changes in the involvement of the different retinal photoreceptors in eliciting brain responses to light. Due to its anatomical connectivity, the amygdala is in good position to quickly receive irradiance information. The medial amygdala receives direct connections from the metalspin expressing RGC in rodents. 23. In addition, a functional pathway linking the retina to the amygdala and bypassing the visual cortex through the superior colliculus and thalamus has been proposed in humans. 59. The hippocampus is connected to the amygdala. 60. And both structures receive numerous afferents from the LC. 61. A potential key component of non-visual response system receiving indirect retinal projections. 35. At present, the functional significance of the limbic responses is unclear. However, it is tempting to suggest that blue light can modulate emotional processing by the amygdala. These effects may be related to the observed positive effects of long-term light exposure regimes in seasonal affective disorder as well as in other psychiatric disorders. 26. Direct assessment of the influence of light on emotional processing should be used to further address this question. Our protocol is very different from those used in vision neuroscience because color vision investigations use isoilluminant stimuli to account for luminescence and brightness brain processing, e.g. Landesman and TSO's 2002, Tutel et al. 2004. The significant difference in left LGN activity between violet and green light onset is therefore difficult to interpret. It is unlikely that it is related to the melanopsin expressing RGC projections to the LGN found in Macas. 25. Since it is not found in the session involving blue light. Conclusion. This study is part of a series of investigations of light processing in the entire human brain. 7-9. We demonstrate that a few tens of seconds of light induce immediate and significant wavelength-dependent changes in brain activity, and that the metalspin expressing RGC seem to provide the most important contribution to these changes. Our results also suggest specific pathways which relay light information from the retina to different brain areas and suggest that light can indirectly enhance the cortical responses by recruiting structures in the brainstem and thalamus. And it's got a list of supporting information, acknowledgments, author contributions, and references. And that's the end of the paper. So this one's interesting. It's earlier than the other papers that we'd read from this author, and so we're getting to see some of the ideas that later it almost taken for granted as they are formed, because this was in 2007. So several years went by between this one and some of these others. It's interesting to see how the brain reacts to blue light. And they were expecting it to react more to green light because that's the range that we pick up. But it didn't work that way, and that's interesting. I wonder what connotations this has to do with like the between the blue and the green light, and as you know, like our ancestors, they lived in a forest, in green plain. Most plant life is green, so we would react to that. We have a wide range of being able to see different types of green because there's lots of different types of green light, and blue light in the sky. You're having that's only blue during the day, so we are active primarily during the day. Might that have had some effect on our development and adaptation? Maybe we we're more aware during the day. So, and it seems like that has to do with the blue light that we're receiving. But maybe that was a development, an adaptation. It's interesting. It does also have some suggestions for practical uses. Like you could have a Kindle or some other reading device, and if you wanted to basically turbocharge or save a part or focus on memorizing a part, you could switch on a blue light filter. That's what I'm saying, and that would stimulate your brain and help you memorize it, just as a tool. And then you wouldn't have that on for the whole thing, but just for the part you're trying to memorize. And so like a definition terms, like doctors, when they, or biology students, when they're trying to run all the different definitions of things as they're reading, because it seems like it has that's what they're suggesting in this paper, is that it can aid in the formation of memories and memory response. So but not even just like the formation of memories, but also like delving into memories. So you could use blue light to help bring up memories. It's interesting. It's very, very interesting. I do like this guy's format of, or him and his team, how they format their papers. It's very well done, and you can see through the references. Like when they did reference past experiments, they didn't use themselves as a reference. They just said, in the past we've done this, this, and this. But they didn't like cite their work like they cited other people's works. So, yeah. If you have any questions on any of this, let me know, and we'll go over that. But that should be it for this paper.